I mean, at first I was worried. I thought, what am I supposed to do for the next 2 to 3 weeks? But then I started playing Animal Crossing New Horizons and catching up on some of my favorite shows, and now I wouldn't mind if this thing lasted forever. But seriously, this'll all blow over by the end of the month. In the meantime, I got trees to plant. Fine, I'll do it myself. Though I may not be the most qualified person to talk on the subject, I did go to college and get one of these. Although calling myself an actual biomedical scientist would be a load of... I do see this as a unique opportunity to share how I would solve this problem. Because believe it or not, I feel like going outside. What you've got here is your typical single-strand RNA virus that's hijacking the receptor ACE2 to get into the cell. All you gotta do is take out the receptor and the virus will have a much more difficult time getting into the cell. Piece of cake.
We tried that.
What's that?
We already tried that.
What do you mean we already tried that?
In mice.
In mice?
Back in 2005.
I'm guessing they all died.
No, they were resistant to infection.
Resisted infection. See? It's like I told you. All you get.
There's more.
What else?
They had overall worse lung function and were more susceptible to respiratory illnesses.
Oh. Because ACE2 is a key enzyme that acts like the brakes of a car when it comes to pulmonary pathogenesis?
Yeah.
Yeah. Okay, I got it. Don't take out ACE2. That's fine. I don't need to. In fact, let's do the opposite. Let's dump a ton of soluble ACE2 into the bloodstream. The virus will be too busy binding to the ACE2 floating around, leaving the cellular ACE2 free to do its regulatory protective thing. It's genius. We did it. I did it. I solved corona. Yes, thank you. Thank you. Oh, thank you. Neil deGrasse Tyson couldn't do it. Bill Nye couldn't do it. Only me, baby. Oh, thank you. Yes. Thank you. Wait. What is it? Won't the accumulation of ACE2 throw off the renin-angiotensin system? Actually, soluble ACE2 has a relatively short half-life of about 10 hours, so no need to worry about any unwanted buildup. This guy. So you give a patient soluble ACE2, and then what? Just wait for the virus to die? A white blood cell will come by and eat it.
What if they're immunocompromised?
Then we'll flush it out with something. I don't know. Look, I'm going outside.
And another thing, you want to prescribe a continuous infusion of ACE2 for days, weeks?
If we start seeing clinical results, yes.
You don't even know how much it costs to make that much ACE2.
No price is too high if it means healing America.
It could take years to collect the amount we're talking.
There is a bacteria-derived ACE2 analog called B38-Cap that could be mass-produced quickly.
Do we know if coronavirus will bind to it?
Maybe. Maybe not.
Do you have any other ideas?
I do, but first, we need to talk about clathrin. Clathrin is a protein that, like my magnetics, likes to form chains. When these chains interact, they form a cage-like structure that is perfect for shaping small vesicles. However, these vesicles can be a virus's free ticket inside of a cell. It happens like this.
the virus binds to the receptor. The cell is confused and thinks, this is something I want to ingest, so it orders the clathrin to begin assembling. The plasma membrane invaginates thanks to the leverage of the clathrin, and in pops a newly engulfed virus. Most viruses infiltrate the cell this way, and so we have developed many strategies trying to stop them. One strategy involves inhibiting the kinases and complexes that make this clathrin connection possible. There are a number of drugs that do this, each with their own side effects, but one of them seems like a particularly good fit for treating coronavirus. Baricitinib is a drug that inhibits endocytosis, like I said, but it also reduces inflammation caused by your immune system. Don't get me wrong, inflammation is good. Too little in any virus or bacteria would cause rampant tissue death. But too much means vasodilation and the accumulation of dead cells, which in the case of coronavirus, means fluid in the lungs and labored breathing. Baricitinib reduces inflammation by diminishing cytokine signals. Cytokines are the immune system's messages. Much like how the body will secrete a hormone to regulate a gland or an organ, cytokines are used by cells to communicate. By slowing down the signal, baricitinib protects against lung injury on top of preventing viral entry. It sounds too good to be true. What about the side effects? Did you follow me here? You said side effects. It says here that 10% of patients got upper respiratory tract infections when taking baricitinib. I thought we were trying to protect the lungs. We are, but most coronavirus patients will already be on antibiotics, so it's fine. We'll just monitor it closely. It also says 10% developed high cholesterol. Not really cured if they still die of a heart attack. All right, enough. This could go on forever. I don't know how to cure coronavirus. I thought I did. But no matter what cellular hack or bio cheat code I throw at it, there will always be repercussions. I didn't even talk about TMPRSS2 inhibitors. They already approved of one in Japan. Or nucleoside analogs. Remdesivir looks promising. Ultimately, it's up to us to determine the best strategy. Now, when I say us, I really mean the actual scientists and the policymakers. You and I can help them, and the nurses, and the bus drivers, and the grocery store folks, by stopping the spread. Social distancing is about to be over, but we still have a social obligation now more than ever to wash our hands and get vaccinated. That's the point of this video. That and to teach some basic virology. Hopefully you learned something. But if you're like me and you want to know more, I've included some sources, in addition to those that I've already cited, that I found very informative. Thanks for watching, and stay safe. Oh, ah. Oh.